What is gluconeogenesis? It is a process in which small and simpler substrates form glucose. Gluconeogenesis runs when the level of glucose gets low in the blood. For example, during fasting. Pyruvate, lactate, glycerol or amino acids are used as substrates in this process. Importance of gluconeogenesis. This process is vital in tough situations because in tough conditions it helps to keep the level of glucose in the blood balanced. It is also needed for acid-base balance. It is vital to form new substances from carbohydrates that are an important part of structural membranes. Location. Gluconeogenesis mainly runs in the liver and kidney cells. Major steps of gluconeogenesis. The enzymes used in glycolysis are also used in gluconeogenesis, but enzymes that catalyze the irreversible steps in glycolysis do not take part in gluconeogenesis. Instead, it uses 4 unique reactions to bypass those irreversible reactions. In the first step, pyruvate gets converted to oxaloacetate. Pyruvate carboxylase is the enzyme that makes this alteration possible. In this reaction, 1 ATP is used and carbon dioxide is added. Let's talk about the regulation of enzyme. This step needs biotin. Acetyl-CoA activates pyruvate carboxylase enzyme. This is the first unique reaction of gluconeogenesis, which is not part of glycolysis. Now the phosphoenol pyruvate carboxykinase enzyme changes oxaloacetate to phosphoenol pyruvate. The second step needs GTP. Therefore, this reaction uses GTP and emits carbon dioxide. This is the second unique reaction of gluconeogenesis, which is not part of glycolysis. In the second reaction, the enolase enzyme makes 2-phosphoglycerate from phosphoenol pyruvate. In this third reaction, 2-phosphoglycerate gets changed to 3-phosphoglycerate. The enzyme that is in action in this reaction is phosphoglycerate mutase. In the fourth reaction, 3-phosphoglycerate makes 1,3-bisphosphoglycerate. ATP is freed in this reaction. Enzyme phosphoglycerate kinase boosts this reaction. Now 1,3-bisphosphoglycerate makes 2 products. The first one is glyceraldehyde 3-phosphate and the second one is dihydroxyacetone phosphate. This reaction raises NADH and releases PI, which is inorganic phosphate. Enzyme glyceraldehyde 3-phosphate dehydrogenase boosts this reaction. In the next reaction, dihydroxyacetone phosphate becomes glyceraldehyde 3-phosphate. Phosphotioisomerase enzyme makes this transition possible. Now 2 molecules of glyceraldehyde 3-phosphate form fructose 1,6-bisphosphate. Remember, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are interconvertible. This reaction converts fructose 1,6-bisphosphate to fructose 6-phosphate. The enzyme in action here is fructose 1,6-bisphosphatase. This reaction is important because it is rate-limiting reaction. Also, it is the third unique reaction of gluconeogenesis. Let's talk about the regulation of this enzyme. Citrate activates this enzyme. Similarly, the inhibiting factors for this enzyme are AMP and fructose 2,6-bisphosphate. The step uses water molecule and emits inorganic phosphate. Fructose 6-phosphate is altered to glucose 6-phosphate. The enzyme catalyzing this conversion is phosphohexose isomerase. This reaction is the fourth unique reaction of gluconeogenesis. In this reaction, the creation of glucose from glucose 6-phosphate takes place. The enzyme glucose 6-phosphatase boosts this conversion. Inorganic phosphate is freed in this reaction and a water molecule gets used.
That is it for today's video. Make sure you are subscribed to the Science Info channel. Like and share this video if you wanna help others. Stay tuned. I will see you in the next video.